Welcome to my channel again. Hello, students. This is Dr. Shanawaz again with the second lecture about the DNA recombinant technology. Second lecture about the DNA recombinant technology. So, in the previous lecture, we tried to understand the basics of the DNA recombinant technology, and we have you might have understood what is the definition of RDT and how it has how can we understand that history of we have tried to understand the history of RDT. And if we will go back to our lecture, we will see that we have found a lot of we come to know about the restriction endonucleases, and then basics of the basic principles of recombinant DNA technology. And now in this lecture, we will now try to understand about what is the recognition site. In the previous lecture, I told you that the restriction endonucleases are cut in the DNA at the specific site, and those specific sites are called as the recognition sites. Now, in this video, I will help you to understand that what is the restriction recognition site and what is this recognition sequence. So, this recognition sequence is a site where DNA is cut by restriction endonuclease. So, we have to first try to understand that that what is this restriction endonuclease. It is the What is this recognition sequence? It is the site where the DNA is cut by the restriction endonuclease. So these sequences are typically short, 4 to 8 base pairs, and are palindromic in nature. In the previous lecture, I told you that palindromic means which can be read from the both sides, and these recognition sequences are palindromic in nature, and they are typically of mostly they are 4 base pairs, 5 base pairs, 6 base pairs, 7 base pairs, or 8 base pairs. That's why we said they are 4 to 8 base pairs. And what is the palindromic? Palindromic means that sequence is read in the same forward and backward on complementary strand. That means we can read the sequence in the forward manner, and in the complementary strand we can read it into the backward manner. So, what are the key recognition sequences? First of all, we will try to understand this recognition sequence, that is AAG CTT. Now, this is the forward strand, that and its complementary strand is 3 prime to 5 prime, and this is 5 prime to 3 prime strand. It can be read in the backward way like AAGCTT. So this means this is a palindromic sequence. In the same way, there is another sequence that is also a palindromic sequence, and this is also the palindromic sequence. So this palindromic sequence is the nature of these recognition sequences. And now, what are the key pointers about the recognition sequence? First key point is the specificity. Each restriction enzyme has a unique recognition sequence. That means each restriction enzyme cuts at its unique recognition sequence. So this is a very important point where you have to remember. This is mostly asked in the NEET exams and other exams. So this very important point, that each restriction enzyme has its unique recognition sequence. So for example, enzyme third, in this Hindi third digestase has this unique recognition sequence, that is AAGCTT, and it uh, this cuts between the A and then, and that at the A and A. That means this is A and A, this A and A. It cuts there A and at the complementary strand it will make cut here and A and A. So this you will see that it has produced a 5 dash protruding ends, that is called overhangs, are called as a protruding ends, and this type of cut is called as a sticky cut. This type of cut is called as a sticky cut. And if you can also see EQRV digest, its recognition sequence is GATATC, and it cuts between the T and A, that is T and A. It makes the cut here, and below it will make the cut at the same side, that is T and A side, and this is called as a blunt end cut. So you may have understood what is the blunt end cut, where there is no overhang, or there is no protruding end. If there is no protruding end, no protruding end, and no overhang, it's called as a blunt end. So this is the sticky end, this is a blunt end. So you have to remember which restriction enzyme, what is the recognition sequence of this restriction enzyme, and what type of cut they make the, either they can make the blunt end cut, or they can make the sticky end cut. So another is palindromic nature. I have explained you what is this palindromic nature. Many recognition sequences are palindromic. For instance, the recognition sequence for EcoR1 is GATATC, and is palindromic because it's complementary. Is also, it can be read as the backward way. If we are reading it as a GATATC, in we can also read it as a GATATC. There is no change in base pair. So this is the palindromic sequence. Though we have understood that the key points about recognition sequence are, the first two is, one is specificity and another is palindromic nature. Now another is cutting pattern. It has I have also made you explain in this first slide that what is the cutting pattern. Restriction enzyme is cut the recognition sites either blunt form, or they can produce a sticky ends. So sticky ends have overhanging single stranded DNA, while blunt end do not have. So this is these are the different restriction and endonucleases, and this explain what are the type of cuts they make. So EcoR1 is from the E. coli, BAM H1 is from Bacillus amylolicufaciens hindi. Third is from the Hemophilia influenza. Not one is from the Nocardia otitidis, and in the same way, SAW 3A is Staphylococcus aureus, and POW third is from Proteus vulgaris, and SAMA is from the Sterratia marcescens. So now uh, you have understood that these are the three restriction endonucleases which cut in the form of the blunt end cuts, and they up above four are not H, no, eco R1, BAM H1 hin third, and not one cause the sticky end cuts. Now the important point is some restriction enzymes with sources and recognition patterns. So these are other types of restriction endonuclease like TAC, Thermus aquaticus, and KPN1, that is Klebsiella pneumonia, Streptomyces, Phycochromagnes, and za XBA1, that is Xanthomas baderi. So these are the other types of the restriction endonucleases and their recognition sites and their sources. Now another point is, when this cut is made, another enzyme is that is called the DNA ligase enzyme. So what is the use of this DNA ligase enzyme? So the cut DNA fragments are covalently joined together by DNA ligases. So its role is joining covalently joining the DNA cut end. That means DNA is cut. If the DNA is cut at any point, so this ligase enzymes joins them. It joins them. These enzymes are originally isolated from the viruses. This is important point, that these enzymes are originally isolated from the viruses, and they also occur in E. coli and eukaryotic cells. So these ligases are I'm saying the isolation of these, they are present in every organism, in every cell, but the isolation is from the virus, and they also occur in E. coli and eukaryotic cells. So DNA ligases work by catalyzing the formation of phosphodiester bond between nucleotides on one strand of the double stranded DNA. So what is the mechanism how they join these cut strands. So they form the phosphodiester bond. This backbone is having the phosphodiester bond, and these are the hydrogen bonds between these. T and A forms the double hydrogen bond, and G and C forms the triple hydrogen bonds, but they are joined to each other by phosphodiester bond. So these ligases helps in joining the phosphodiester bond between the nucleotides. So DNA ligase is able to create the covalent linkage between 5 and phosphate group of one chain with the adjacent 3 OH group. So this covalent linkage helps us in the formation of the ligation bonds, and in this these cut DNA join together. The reaction is not important only to join the nucleotides during replication, but also repair DNA damage. And at that time, if there is a DNA damage, and at that time these DNA ligases help in the joining of the two DNA. So this is the 5 prime end of phosphate, and this is the OH end of the 3 prime end of OH, and this in presence of ATP they helps in the formation of phosphodiester bond, that is the normal linkage, and this is the enzyme catalyzed ligation. This is the enzyme catalyzed ligation that you have to remember, and it needs ATP for its formation. It needs ATP for its bond formation. Now another other enzymes which are very important for the in during for to perform the recombinant DNA technology, or for recombinant DNA technology, are alkaline phosphatases. The enzyme alkaline phosphatase helps us in removal of terminal phosphate group 5 and prime, and it prevents self annealing of the vector DNA soon after the cut DNA by restriction endonuclease. So, what is the work of this alkaline phosphate? It helps in the removal of terminal phosphate group from 5 and prime. If there is no phosphate, so this ligation may not help. Yeah, self annealing in the vector do not take place. And then another is ball 3-1 nuclease for progressive shortening of the DNA. If we want to cut the DNA progressively, we need this ball 3-1 DNA nuclease. Another enzyme is DNA polymerase. It synthesizes the DNA complementary to the DNA template. Another enzyme is DNases. They produce the single stranded nicks in the DNA. These are the questions that are mostly asked in the examination, and you have to take care of these all enzymes. Remember all these enzymes. So DNases are produce a single stranded nick in the DNA. Exonuclease third, it removes the nucleotides from the 3 prime end of the DNA. That is 3 prime end of the DNA. Now there are, if there are nucleotides present at the 3 prime end, we need the exonuclease third. Uh, Now there is delta exonuclease that removes the nucleotides from the 5 prime end. So polynucleotide kinases transfer the phosphate from ATP to 5OH from end of the DNA. Restriction endo enzymes cut, we know these cut the DNA with a specific recognition site. Reverse transcriptases is used to synthesize the complementary strand from mRNA. It is also known as RNA dependent DNA polymerase. It is very important. You have to remember this. Right? What is the second name of the restriction transcriptases? And it all isolates from, it is isolated from the retrovirus. RNase A cleaves and digests RNA and not DNA. So when you have to perform the isolation of the RNA from the blood or any other organism, so you need to make everything RNase free. So what type of RNase is that? It is RNase A. Now, RNase H cleaves and digests the RNA strand of RNA and DNA heteroduplex. If there is a heteroduplex, that is RNA and DNA are joined together, they form a duplex. At that time, RNase H is used, and it is done mostly when there is reverse transcription. Now we have to remove this RNA from that DNA, and RNase H is used. TAC DNA polymerase used in polymerase chain reaction. S1 nuclease degrades the single stranded DNA and RNA. Terminal transferases add nucleotides to the 3 prime end of DNA and RNA and useful in homopolymer tailing. That means if we want to add the AAA to the DNA strand or AA to the RNA strand, and that is called as a homopolymer tailing. and it is terminal transferases. That means they help in forming this homopolymer tailing. This if this is strand of the DNA, then we want to add A to it. That is called as a homopolymer tailing. Now, uh, we have another topic now: host cell. What are the host cells? And the fact they are called as the factories of cloning. The host cells are the factories of cloning. That means if you have you got, for example, now we know there is a vector and we cut this vector, it is open now, and we got the gene of interest. We will insert this gene of interest into this vector, and now this gene of interest is in the vector, join it to the vector. And now who will clone this vector now? For that we need the host cell, and this host cell, this vector will be introduced into this host cell, and this host cell will help in the cloning of this vector, and it will form the multiple clones of this vector, and that is why they are called as the factories factories of cloning. They are called as the factories of cloning. Now, host cell are the living systems of or are cells in which carrier of recombinant DNA molecules or vectors can be propagated. That means the carrier of the recombinant DNA molecule or vectors, whatever is the carrier of that recombinant DNA molecule, it is to be propagated. We have to multiply it. Now, the first host cell is a prokaryotic host cell. It is E. coli, was the first organism used in the DNA technology experiment and continues to be host cell of choice. So drawback: in E. coli cannot perform post-translational modification. This is the first drawback. If we want to perform the post-translational modification, it's not possible in this host cell. So host cell strains are DH5 alpha, BL21, DS3, and JM109. So these are the different types of host cells. For example, if you anyone will ask you that where you have done the cloning, you will say yes. So he will ask about the in which cell, bacterial cell, you have performed the cloning. They will say that we have performed this cloning in the E. coli, and he will then ask you the which type of strain it was. So you have to remember what type of strain was we have taken for the cloning. Is it DH5 alpha? It is BAL291 and JM109. Bacillus subtilis is a rod-shaped non-pathogenic bacteria. It has been used in the host industry for production of enzymes, antibiotics, and insecticides. Eukaryotic host cells are preferred to produce the human proteins, since these hosts with a complex structure are more stable to synthesize the complex structures and are more stable to synthesize the complex proteins. If we have to perform the cloning of these complex proteins, so we need the eukaryotic host cell. And most commonly, eukaryotic host cell are yeast, Saccharomyces cerevisiae, and tissue plasmogen activator synthesized by mammalian cells. Other organisms and cells used for genetic engineering purpose is Saccharomyces cerevisiae, and eukaryotic post-translational modification happens there. Well characterized, easy to culture, and high transformation efficacy. This is Pichia pastoris is high yielding proteins. If we need high yielding proteins, we use Pichia pastoris. Pastoris can produce large amount of recombinant proteins. Strong promoter and uses strong inducible promoter like AOX1 for high level of expression. Why is it used for recombinant proteins? Because it uses a strong promoter. We can perform the post translational modifications in it, and also it efficiently secretes recombinant proteins, simplify in purification. Now there is some problem when we have to we performed the cloning, but we need the recombinant protein, and we there are most of the cells we face the problem of purification. But in case of pastoris, we perform the purification simply. Now this is Chinese hamster ovary cells. uh, they perform the complex modifications such as glycosylation, and widely used in production of therapeutic protein and monoclonal antibodies. So these Chinese hamster ovary cells are the host cells, or we can say who these, they act as a host cell in recombinant DNA technology. Regulatory approval, many Chinese hemisphere host cells, CHO cells derived products have received regulatory approval and making it easier for new products to gain approval. So another is human embryonic kidney cell, and this is derived from human cells and providing more human-like environment for protein expression, high transfection efficacy using various methods, used for transient and stable expression of proteins. And insect cells are SF9, SF21, and Hi5. And bacillus system is efficient expression system using bacillus vectors. And post there is also this post translation modification can take place in these cells and capable of producing high yield, high level of recombinant proteins. So in the next lecture, this is the end today, we will complete here a short video of 20 minutes, and in the next lecture we will try to understand what are the cloning vectors and what are the different types of cloning vectors. So keep watching this channel, subscribe this channel, and support this channel, and share all these videos. Thank you, all of them. You can point out the questions, and if you have confusion, we can discuss into the next lecture.